Well, thanks everyone for joining. This is our end of year Enigma year in review for 2025. We really appreciate you joining us at a very busy time of the year. This call's a little bit earlier. It's at 10 a.m. Pacific because I'm currently in northern India, in Chandigarh. So before we begin the remainder of the call, I know there's a few words that Dr. Odile van den Heuvel and Nick Van Wey and Ninka Grunewald are going to say at the beginning of the call. Odile, are you ready to say a couple of
Yes,
words?
yes, please. Can you show me the next slide? Yes, so it's a bit difficult, so I wrote down my words. So just over a week ago, Professor Dan Stein passed away after a very short but relentless illness, and finding the right words is really difficult. With Dan, we have lost a remarkable and richly diverse man. And from all the messages that we got last week, it's clear how important Dan was in his work and lives of so many people across generations. And so some people know him already for more than 40 years, other people only met him indirectly, but everyone who crossed his paths found, his enormous dedication to psychiatry and to global mental health. And from his base in Cape Town, then connected the whole continent, Africa, that is so often underrepresented in science with the rest of the world. And he felt very responsible to keep issues relevant for Africa high on the global agenda, like maternal mental health, HIV, and addiction. And at the same time, his own academic roots were firmly based in OCD and anxiety research. And within ENIGMA, he co-founded the ENIGMA OCD Consortium with me and the ENIGMA Anxiety Consortium with Nienke, Janne-Marie, Nick, and Danny. Dan had a deep and generous love for science in its all diversity, from genetics, imaging, epidemiology, diagnostics, treatment. And that came from more reflective perspectives to very testable hypotheses. And he really cared about the next generation. So many people know him as a mentor, and he really wanted to help them finding their footing in the field. And he was a very approachable and supportive mentor to so many people. And then strongly believed in team science, and time and again, he was the quiet force connecting people who didn't know each other and who started strong collaborations. He was a sharp-minded but very gentle, both in his word and his acts, always thoughtful and strategic. And through this kindness and authentic connection, I think he made many things in motion. He loved literature, philosophy, and by this way, he also got different perspectives on so many topics. And then really wanted to continue contributing to the field. He kept working until the very last moment on all the different topics that he found so important. So I thought, let's honor him by continuing his work and following his example. And I thought, I got 5 lessons. So the first one is work hard. I think we all do that. The second, help the next generation to find their way. The third, collaborate with kindness. The fourth, build the bridges between the continents, the topics, and the disciplines. And five, stay sharp in mind while always gentle in the connection. And let us also give him something back. So let's gather our memories or pictures and let's make a book for him. I promised his wife, Heather, that we as the Enigma family will bring together all our memories so that they can also hear what he meant for the field and for all of us. I think that's what we can do to give something back. So, yeah, thanks a lot, Dan, for all you did.
Thank you so much, Odile. And I know Nick and Ninka also have a few words to say.
Yes, I think, yeah, thank you. So on behalf of the ENIGMA Anxiety Working Group and my fellow coordination team members, Ninka and Jana-Marie, I would also like to say a few words to remember Dan and his contributions to ENIGMA as a whole and ENIGMA Anxiety in particular. And I was already alluded to by Odile, Dan was not only the colleague and friend you wish for, but as many of you know, also an inspiring scientist, mentor, visionary, and someone for who true collaboration was the key to success in life in general, but in particular in science. So inspired by the success of ENIGMA and ENIGMA OCD, Dan and I started the adventure of ENIGMA Anxiety now a decade ago. And Dan was very keen on immediately involving promising young scientists, Nienke and Anne-Marie, in a coordination team as we aspired to build a collaboration that could continue to prosper without us. Sadly, that moment arrived too early for Dan. We're going to miss him and will continue ENIGMA Anxiety in this spirit. Thank you.
Thank you so much, Nick. And Ninka, did you have a few words to say, or
yeah,
yes,
do go ahead.
I do. I wrote some personal words. In 2015, I came to UCT to work with Dan Stein. It was proudly part of my introduction for the first few years as part of my professional identity. Hi, everyone. I'm Nienke Groenewald, and I work with Professor Dan Stein at UCT. When I started, Dan asked me to sit in on the ENIGMA meeting and take minutes. The next meeting, he asked me to create an agenda. The next meeting, he asked me to coordinate a group. Before I knew it, I was in charge of the ENIGMA contributions from UCT. This illustrates key aspects of Dan's mentorship: the ability to direct people into roles that they are strong in, the ability to set big goals, and the ability to convince others to do things that they may not even have thought possible themselves. When I attended a K43 grant writing workshop, I was advised to switch mentors, as after 2 to 3 years, you would have learned everything that you could possibly learn. But in the past 10 years, I've moved from postdoc to junior fellow that I did with Dan's wife, Heather Saar, who is also a highly accomplished clinician scientist, to associate professor with Dan's support. And at every stage, I was able to learn new things from Dan. From science writing to supervision, research line and strategies, he covered it all. Dan always had a keen eye out on opportunities for his staff and students to grow and to build capacity. Locally, Dan founded the UCT Brain Behavior Initiative, and I included a picture here of the local leadership, which laid the groundwork for the UCT Neuroscience Institute, where he was the inaugural scientific director. I am only one of so many people that Dan has mentored for over 10 years, and many of his mentees have moved on to become international leaders themselves. Dan truly loved the collaborative spirit of ENIGMA. He was passionate about enhancing African representation in global neuroimaging, in genomics, and in neuroimaging genomics research. As someone from the global south, he advocated for equitable partnership. He did not see us at UCT only as data providers, but he also saw us as co-leaders. He insisted that ENIGMA members at UCT would lead their own projects, most importantly in the areas that matter in Africa, that is HIV, substance use, maternal and child mental health, and more. In this way, he brought the global mental health movement that he was a key part of and the values that he held dear into ENIGMA. That, to me, really is the most important thing that I could learn from Dan. With everything that he did as a researcher, he intended to make a positive impact on this world. And when I look at the outpour of so many heartfelt messages of inspiration that have flooded the ENIGMA email list in the past week and that reach far beyond it, it is clear that he has achieved an immeasurable impact in his life. To end with, I wanted to reflect that when the goal for impact is the starting point, then our work is meaningful. And today we can be so grateful for the heaps of inspiration that Dan has left us with, and we can continue the work that he found so meaningful together. Thank you.
Thank you so much, Ninka and Nick, and also I know you speak on behalf of Jana-Marie and Aaliyah and the Cape Town team. We're hugely indebted to Dan. I mean, he was a giant among men. We're indebted to him for all the hard work he did for us. We used to say in Los Angeles that Dan is everywhere all the time. And so we'd see him far from South Africa, far from Los Angeles. He'd cheer us up, connect us to other people, connect us to new friends, create opportunities for us. And he was immensely proud of his family. He was immensely proud of you, Ninka, of Aaliyah, of all of the Cape Town team for all that you've achieved. And there is a line in Thucydides saying, "The whole world is a sepulchre for famous men, for it is not only the memorial in their native land that commemorates them. In foreign lands far from their own, their vision lives on in all the inspired. Their vision lasts forever and is written in the hearts of humankind." And that is very true of Dan. And all we can really do is carry on with his vision, his creative vision in our professional lives. And we'll listen to Dan's family and the Cape Town team to best commemorate his remarkable life in the fullness of time. So thanks very much. Okay, so for the next 55 minutes, we will give our customary end-of-year overview of ENIGMA. And there's, despite it being a very sad day, there's a lot to commemorate today. There's new grants, new working groups, new publications, major methods developments in fMRI, tractometry, and normative modeling, updates from genetics, GWAS, copy number variation, polygenic scores. And we'll give a lot of updates from clinical working groups. And it's a real privilege to do this every year. I know Sophia and I and the USC team receive all of your updates from psychiatry, neurology, brain development. This is the first time that we're seeing all this data together. And there's so many creative minds here that you'll have excellent ideas. And we'll finish with a wrap up on workshops and training initiatives for ENIGMA and upcoming opportunities to meet in person. So here's ENIGMA, as you all know. Now we've reached 200,000 data sets across all modalities in 47 countries. And here's the latest diagram of the groups at the top there focusing on methods, and then in the middle focusing on psychiatry, neurology, and brain development. There's been a lot of highlights this year. So the CINP President's Brain Health Award went to Dr. Lian Schmal, who leads ENIGMA MDD. There was a terrific meeting in Croatia led by Sharash Eka, Ali Safid Konal, and Kursat Altinbas. There was a wonderful meeting on anorexia at IGC where Caitlin Lloyd, Laura Berner, Clara Moreau, and Joanna Steinglass attended. And we've had great visits from Willem Bruin and Johanna Bayer, many others throughout the year that we're very, very grateful for. There's been meetings in India. So the NIMHANS team, who many of you know, led by John P. John and Ganesan Venkata Subramanian, have led a tremendous initiative that has been collecting data from 200 elderly people with Alzheimer's, MCI, healthy aging, and a whole host of genomics and omics assays, actigraphy, social and environmental determinants of health, diet, access to healthcare, to understand factors that influence brain aging and mental health in India. There was a huge showing from ENIGMA at SOBP. Dr. Carrie Bearden was awarded the SOBP Gold Medal in Toronto. She's now the SOBP president for 2026. And there was a number of awards. Congrats to Ian Gottlieb, who was awarded the Global Open Science Best Paper Award. This was a get-together in Toronto, a lot of fun. An even bigger get-together in Australia for OHBM. And here's the social event. I do want to recognize Amy Brotman, who won the Mentor Award at OHBM, Jack Van Horn, who won the Education Award at OHBM, and Dr. Neda Jahanshad, who's the Associate Director for ENIGMA, won the Open Science Award at OHBM in Brisbane. So congratulations to all of you. There was an opportunity at OHBM to give the plenary. So the OHBM Telerac lecture was a summary of all of ENIGMA that we can send to new students and new members and trainees on how ENIGMA's neuroscience results fit together and some emerging cross-disorder principles. So just a quick wrap up of where the grants are at the moment that support ENIGMA. There are 34 active grants. There were just awarded grants to Sophia Frango on the Centile Brain Normative Modeling System, to Neda Jahanshad and Claudia Barth on menopause and its relationship to aging. Shiju is studying PTSD, MDD, and anxiety, and OCD for that matter, in a new R01 from NIH. And congrats to Yella Lamsma for winning a Vaney grant studying the correlates of aggression in a variety of imaging modalities. This leads us into new territory methodologically. Shiju, represented here by Juan Maya Torres, is looking at deep learning for subtyping of neuropsychiatric illness using very, very novel methods that read in functional and structural brain imaging data. Currently in review, and good luck to the people who have grants in review, is an fMRI grant led by Clara Moreau and Frank Hillary, a couple of Origins grants looking at the brain in infancy, ENIGMA Pakistan, that is being led by Maheen Adamson and Mohammed Parvaz, that is under review at the Wellcome Trust, and a number of other efforts, either looking at multivariate patterns in psychiatry, one led by Peter Kochanov, and a binge eating initiative led by Laura Berner. These are the plans to resubmit. I won't go through all of them, 10 grants. And as NIH enters 2026, we all need to look at this carefully. There are a couple of new mechanisms that allow non-US sites to be on NIH grants. There's now a maximum of 6 submissions a year. As PI, pay lines have been abandoned. That's the pay line cutoff for funding. Only the top third of grants are getting reviewed, and there's also ongoing disruptions and delays. But despite these shutdowns, there have been some successes. Now, this list of publications, 39 in total for the year, I won't read them all out, but thank you to Sophia for putting links in. I'd encourage you at the end of the call to click on these. There are links to all of the major publications of the year. There are 12 additional that are being submitted or revised. Congratulations to all of you who have had publication successes. And there are also new working groups. So this remarkable paper by Philip Homan and Finn Rabe led to the suggestion that there be an ENIGMA group on the relationship between the retina and the brain. So it turns out that cortical thickness and macular thickness in the eye are altered in schizophrenia, bipolar, and major depression with an effect size that is actually quite remarkable. So you'll remember the ENIGMA maps for these 3 psychiatric conditions, and there is an imprint, as seen here in this paper by Foyos Gorgiadis, an imprint in the eye as well. So they're interested in setting up, with any of you who are interested, a group that relates normative differences in the eye to brain metrics and clinical metrics in these disorders. So if you have an interest in collecting images of the retina, or do reach out to Philip Homan and Finn Rabe. There's a new group on cerebral small vessel disease, led by Matt Lennon, Parminder Sachdev, and colleagues. This is going to be looking at the effect of cerebral blood flow and restrictions of it to brain aging. And if you're interested, fill out a short survey. Karen Kayenbergs and colleagues are looking at ultra-low field MRI. This is a type of scanner that can run off the mains in the wall, a 900-watt scanner at very, very low field. And if you have this type of data, do get in touch with them. They're interested in merging data at low field, which also can be obtained in under-resourced settings. There is interest in setting up a working group on functional near-infrared spectroscopy, which is a brain imaging technique that's portable. It shines near-infrared light into the brain and tracks blood flow. And if you are interested in that type of modality, have a chat with Jackie Stevens and colleagues. Jessica Roy and Sophie Volk have started a group relating social abilities and social environments to brain structure and function. They are interested if you've been administering questionnaires on empathy, compassion, social responsiveness, social cognition, theory of mind, emotional and affective skills. See a survey if you want to take part. If you have whole blood mRNA, have a chat with Ida Pivandi. They're interested in relating transcriptomic markers to psychiatric markers and imaging in particular. And also ENIGMA language. This is very, very encouraging. If you have data on whether your participants are bilingual, there is a vast study now being led by Olga Kapinska, which is looking at the relationship between brain aging and mental health and knowing several languages. It's a wonderful study that they've put in a registered report for, and 33 ENIGMA sites have contributed to it. ENIGMA language has a branch on developmental language disorder led by Gail Doucette in Nebraska. If you're interested in DLD and brain, have a chat with her. And there's also a group led by Christian Hyde and Ian Fulcher at Deakin University in Australia, looking at developmental coordination disorder, which is a disorder of movement, and also co-occurs with neurodevelopmental conditions such as ASD and ADHD, and have a chat with them if you're interested. Max Landsma and colleagues have started ENIGMA Tremor, looking at gait problems and its relationship to cognitive decline, which is of great interest because of its relationship to Parkinson's and other disorders. Valentina Lorenzetti has started a group on experimental cannabinoids. Have a chat with her if you're interested in that. And we'll talk about some new methods. So here is Dr. Jan Beitelaar, who isn't on the call because his son is getting his PhD today. Congrats to Jan's son. But look at this T-shirt. So you can see all of these maps. You can find them in the ENIGMA toolbox. In fact, if you look at the maps of different brain disorders and their effects on structure, diffusion, and resting state, you will notice that 3 primary workflows are run by ENIGMA working groups, except for a couple of them that focus on EEG, MEG, and spectroscopy. And a number of observations have been made. So this is work by Rebecca Cooper, working with Maria Jalbrzykowski and David Glan. And she observed that the more prevalent disorders in psychiatry and development are associated with smaller brain abnormalities, and the less prevalent, the rarer disorders are associated with greater brain abnormalities, the y-axis being the Cohen's d, or the effect size between cases and controls. And this is an example of a study, a very interesting study, that has been made possible by all of the ENIGMA groups working together. And of course, this can be done with DTI. 18 disorders have been studied with diffusion tensor imaging, with protocols developed by Nadezhda Hanshad and Peter Kochanov. There is a new DTI t-shirt as well. And more recently, functional MRI has been, a workflow has been developed by Clara Moreau, Leah Waller, Ilya Veer, Frank Hillary, Tomasz Fodran, and Lynn Belek. And this is now being used across over 25 disorders to compute atlas-based connectivity, canonical brain networks, even perfusion and gradients from resting state functional MRI. So here's a diagram of all the methods that are being used. Within yellow are the main ones. I will add that you can go and find them on the ENIGMA GitHub if you ever want to know where these are. And some of the less commonly used methods are also gaining popularity. So if you look at brain age, over 25 studies have looked at brain age, which is the apparent difference between your brain's chronological age and how it looks. There is going to be an overview paper by Gilles de Grey and Laura Hahn. I mean, just look how many ENIGMA groups have been looking at brain age. It'd be very interesting to see what the commonalities and differences are across conditions. The fMRI workflow has been led by Clara Moreau, Leah Waller, and their team, Ilya Veer in particular. And this is now used at 81 universities across 22 countries. It's a processing workflow that's based on fMRI prep, and an enormous achievement has been reaching consensus across ENIGMA in what to compute. So if your working group's analyzing resting state fMRI, please follow these guidelines, make your analysts aware of them, please send one analyst per working group to their functional imaging calls. And if you want to listen to the decision process as to how this workflow was arrived at, there's a very detailed description of it there. It will compute for you all sorts of functional metrics on the synchrony of different brain regions, measures of perfusion, and dynamic fluctuations in the brain. And they are beginning to write a publication in conjunction with a vast team of people who are QCing data across the world to quantify the best metrics, including a tool called WonkyCon, which will identify which workflow is best at identifying differences in your data set. A huge thank you, thanks to Clara and team for putting this together, to all of the people who are doing quality control on resting state fMRI across the world. These data sets are invaluable for testing the different workflows that they're developing and coming up with conclusions about the best methods to use. And they're putting together a paper that describes all of this. They are planning to hire new developers, and they're also going to reduce the processing time and eventually add task-based fMRI. So congrats to Clara, Leah, Ilya, and team for this vast array of 28 working groups now using half-pipe to process their functional data. The PTSD workgroup was the first to have a large number of fMRI projects, and Dylan Sun and Nick Steele and colleagues developed IBMMA, which is a unified package for statistical analysis of fMRI. Now, there's a number of really interesting papers about functional connectivity in PTSD. Ceci Hinojosa, Seth Disner, and Dylan Sun have been reporting on alterations in seed-based connectivity of the amygdala and hippocampus in PTSD. And of methodological interest is the software they develop. So Dylan Sun and Nick Steele have written image-based meta and mega-analysis. Now take a look at this. This is really useful for everybody. It will take your files, your brain images as inputs, and the models that you specify. It will output maps, tables, various figures, all outlined in their paper and in the GitHub at the top. And it will give you tables of how long it took to run and all the statistical maps that could go into a paper. And congrats to Dylan and Nick for creating this. It's very, very, very helpful. In addition, because it's complicated, it will show the proportion of subjects with missing data at each box in the brain. how classification degrades with larger and larger fractions of subjects thrown out. And all of these things are helpful in identifying any aberrant data in a very large-scale study, such as the kinds that we do. In addition, there are 16 active projects using fMRI. Congrats to Hugh Garavan and Anthony Giuliano for processing vast numbers of data sets. They're now starting to process task-based functional MRI from abuse of alcohol, cocaine, opiates, cannabis, and nicotine. Frank Hillary is looking at a famous hypothesis in brain injury, that there is early hyperconnectivity in hubs of the brain, followed by posterior loss of hubs, and then hypoconnectivity in the chronic phase. As this project goes forward, the pipelines are being analyzed in detail. Melody Kang, working with Chris Ching, has been looking at brain synchrony or brain functional connectivity in bipolar disorder, and they are actively testing how different settings and workflows, in particular harmonization methods like block combat, influence the results. And this data shown here has just been sent to OHBM, shows that combat seems to pick up signals better than standard random effects modeling. In this study, it's a study of FALF, of low amplitude fluctuations, which are altered in bipolar disorder. Also, Nick Livingston, working with Gemma Medinos and Matthias Kirchner, have been investigating schizotypy, and they have found that spontaneous fluctuations in brain activity occur at rest in schizotypy. They have a truly vast sample where high positive schizotypy was associated with low FALF fluctuations of the posterior cingulate, which is consistent with findings in clinical psychosis. Aliyah Ibn Idris Al-Siddig from the University of Cape Town with Dan Stein and colleagues have been examining HIV. And perhaps in good news, I think Dan would be happy to see this news, there were no differences in the HIV, people living with HIV versus healthy control. So perhaps antiretroviral therapy is protecting their brains from deficits. Congrats to Aliyah and her team for this remarkable study. In OCD, Nadia Ginalia and Dr. Vandenheuvel have been looking at prediction from task-based fMRI of outcomes to cognitive behavioral therapy. Now, these are quite significant effects at the group level. It's more tricky to predict outcomes at the individual level, but they've been using machine learning to do it. And in a similar vein, Joaquim Radoa and Dr. Fulona have been looking at fear conditioning. Now, for those of you who don't know what it is, it's used to study conditions such as PTSD that are marked by pathological fear. People learn to expect something unpleasant when they see one cue, but not when they see another, such as an electric shock or something given when a stimulus is. And the difference in brain response reflects fear learning. And they found that the areas of the brain that are engaged during fear conditioning, that are more active in the conditioned stimulus than the non-conditioned stimulus, are in the salience network. And this is highly interesting. It argues for threat anticipation as part of salience processing in the brain, which is a classic component of PTSD. This immense toolbox developed by Neda Jahanshad's team at USC, in particular Iyad Bargari, Bram Chandio, Wendy Feng, and JD Alibrando, and a huge team. They meet every Thursday for hours on end, and talk about how they've tested this on many, many data sets across the world. They don't want to release it until it works, but look out for this in the coming weeks. This white matter lesion quantification tool developed by Sarah Albachari and Hamid Haroun has been released as a Docker. It's very, very useful for identifying white matter lesions. And this huge work of software by Yang Yim and Chris Ching is for 3D shape quantification. It was developed for ENIGMA bipolar, but is quite useful for any data from any group that's represented as these mesh models. Huge kudos to Dr. Sophia Franco, Riange, Shalala Haas, and their team for creating Centile Brain. This is a normative framework that will assess deviations in new individuals relative to an enormous population reference that has been generalized throughout the world on diverse data, which all of you have contributed. You can use it without data transfer, which is part of why it's become so popular. 6,000 users worldwide have used it. This now will norm your structural data. They're going to be releasing resting state normative models and even white matter connectivity normative models soon. If you have repeat MRI data, longitudinal data, please get in touch with them. That's very important to test whether these models are consistent over time, and even develop models that will assess whether a brain change is normal or not. If you want to harmonize your DTI, Alyssa Zhu and Neda Jahanshad have software for you. You can see the rather messy data on the left has been repaired on the right and made into a nice age trajectory for different microstructural measures of the brain. If you have diffusion data from healthy controls, contact Julio Villalon. He's topping up a normative model. The lifespan changes in microstructure have been charted here. You say, well, why do you need more data? There's already 50,000 people in this. But the people in the outer quantiles are the ones that are most interesting to us, and we want to fill out the amount of data that we have there. Eagle Eye, developed by Evelyn Deutscher and colleagues, will allow you to QC data in people whose scans have brain lesions. Very, very important work if you're looking at stroke or, for that matter, brain trauma, and have a look at their advanced guide for identifying errors in parcellations in people with brain lesions. Matt Kempton's team has been publishing with voxel-based morphometry. It's an assessment of throughout the brain the areas of differences in gray matter, white matter, and CSF density, this latest report being in PTSD. Shane Prakar and others have published on bipolar disorder and OCD, and you'll see as the number of data sets increase, the results coalesce into an interesting convergent pattern. Alison Nugent, Alan Amist, and Jeff Stout have been extremely productive this year with a number of papers processing 33 MEG magnetoencephalography data sets across the world. You'll see that this processing pipeline that they've finished, it's now under review, has been used to make normative charts. So these are measures of brain metrics from MEG, a very versatile imaging technique that can be used even in children who are moving around. And they have now lifespan charts that can be used to calibrate things like delta power and other metrics that are of interest in patients. Brenda Bartnick-Olson and Alex Lin and colleagues have created a pipeline for spectroscopy. It's already being used for voxel co-registration and segmentation. If you're interested in their normative project, do reach out to them. Fabrizio Pizzagalli at the University of Torino in Italy has been working with his team on sulcal analysis. So this could be of great interest no matter what disorder you're studying. They have normative charts here for central sulcus width. They've been looking at neurogenetic syndromes such as 22q11 deletion syndrome and found that these metrics are actually rather sensitive to changes that might not otherwise be apparent. And thanks to Faisal Rashid, who used to be at USC and is now in Lucca in Italy, for developing a workflow to harmonize this sulcal data across sites. Nikhil Bhagwat, Sebastian Urchs, and J.B. Polin developed a system called NiPoppy and NeuroBagel that has been piloted to organize and process data in ENIGMA Parkinson's and ENIGMA Tremor. Now, just to say what this is, and this has been provisioned across the world, J.B. Polin at OHBM gave a talk on how methodological variation accumulates throughout a study, and their system, which is called NiPoppy and NeuroBagel, allows you to annotate all the clinical and demographic data according to a standardized dictionary. And essentially, if you're leading a working group on a disorder, it allows you not just to organize the covariates, but it allows you to query which data sets have different data sets, and it provides an entire informatics system to manage and organize the multi-site data. Moving to genetics, congratulations to Vilta Baltramaitita, Elster Walton, and Danai Dima for their genomic screen of brain age. Now, brain age, again, is the apparent aging of your brain on MRI versus your chronological age. And they found 32 genetic polymorphisms that are influencing brain aging. And their Mendelian randomization results show a potentially causal influence of high blood pressure and smoking initiation on accelerated brain aging. Nadine Parker and Ulla Andreasen have been combining polygenic risk scores for bipolar disorder and imaging risk scores for bipolar disorder, showing that the combination outperforms each separately as an indicator of whether someone has bipolar. A truly vast study by Shuyi Shen, Heather Worley, and Andrew McIntosh and Enigma MDD looked at polygenic risk for depression in 50,000 people. They found that people that have a high polygenic risk score for depression have lower surface area in many of the orbitofrontal and limbic areas that are implicated in MDD, but no effect on cortical thickness, which essentially makes you think that the thickness changes in MDD may arise not from genetics, but from later life effects, either chronic stress or medications or illness progression. Other studies of polygenic risk for disease include this study where Nushina Basi and Ellen Dagger and ENIGMA PD showed that people with high polygenic risk for Parkinson's disease, but who do not have Parkinson's disease, they already have significant cortical changes. You'll see that the imprint of carrying genetic risk for Parkinson's disease looks a great deal like having Parkinson's disease. So they have a subsignature of the disease. And in similar work, Boris Bernhardt and colleagues, including Alexander Gno, have found the same thing in epilepsy. People with high polygenic risk for epilepsy have a subsignature that resembles the atrophy pattern in people with epilepsy. And Enigma Epilepsy, in a project led by Sanjay Sisodia, are looking at the relationship between carrying a lot of SNPs associated with epilepsy and accelerated brain aging as measured by brain age. Enigma Plasticity, led by Rachel Brouwer and Hilke Hulshoff-Paul in the Netherlands, constructed age-dependent polygenic scores for longitudinal brain change. Now, what does that mean? It's a combination of genomic markers that will predict how fast your brain is aging, which has been derived from their analysis, GWAS analysis of people who've had several MRI scans. Leah Waller and their team have done a genome-wide association study of task-based fMRI, and they've discovered the first genomic loci that influence the brain's response to emotional phases, working memory, reward, anticipation, and outcome. Congrats to Leah. She's remarkably just finishing her PhD with this incredible paper, and congrats to Ilya and Suzanne. And Henrik Walter has the best excuse for not attending this call. He's getting married this week. So congrats to Henrik. Enigma's DTI genome-wide association study led by Neda Jahanshad now has over 80,000 data sets. If you're interested in taking part, please reach out to them. Dirk Schmidt and Philip Jowinski have completed a genome-wide screen of EEG, measures of brain synchrony, and found that the loci in the genome that influence brain synchrony on EEG are also related to physical and mental health, risk-taking in education, and to the thickness of the cortex. They also have built a normative model. Their target is 30K subjects, and that will be an interesting extension led by Danai Mitseya. They're also looking at GWAS of theta-beta ratio, connectivity, and oscillation dynamics. Enigma Epigenetics, led by Sylvain Derivière at King's College London, has discovered that methylation of homeobox genes is associated with variations in cortical thickness. Now, those of you who remember your developmental neuroscience classes in graduate school will remember these are famous genes. In fact, in mammalian embryonic segmentation, these are marker genes. And so this is really a new lead combining data on epigenetics across 13 international cohorts. And they're finding that methylation at CBG islands is linked to regionally specific cortical volume alterations in the cerebellum and the inferior temporal cortex. Anna Silva and David Linden made an observation about rare genetic variants. They found that how dangerous the rare genetic variant is in terms of the risk for psychiatric and developmental disease is linked to thinning, particularly in the visual cortex. A very interesting observation that the copy number variants that affect the visual cortex are among the most dangerous, and you can look at their study there. It's a very, very interesting link between disease risk and deletions and duplications in the genome. Now, what are the emerging themes across all of these disorders? We've talked about individual disorders. What are the emerging themes across them, and what are specific to different conditions? Wei Cheng and Yuchao Jiang asked how similar is schizophrenia worldwide in 41 cohorts. They began working at Chinese sites and generalized their work to a large number of data sets across the world. And they observed that there are two subtypes of schizophrenia. They used a method called subtype and staging inference, or sustain, and they found that the majority of patients with schizophrenia have a pattern of progressive atrophy that originates in Broca's area, whereas there's a second subtype. This is the first one that is originating in the frontal cortex, and it proceeds posteriorly as the disorder progresses. Whereas there's a second subtype that is possessed by about 38% of the patients in their study, in which the initial hippocampal and temporal atrophy then proceeds anteriorly into the frontal cortex. Now, you could say, what does this mean clinically? They found that there are distinct differences in depression, negative affect, and anxiety only in one of these subtypes, and this leads to great interest in whether medication response might differ between the subtypes. And that was replicated across non-Chinese European cohorts and across their study in China. Now, ENIGMA Parkinson's has also defined clinical stages based on Hearn and Yahr scores. So let's look at this data. The spread of tau pathology in Parkinson's is indicated by cortical thinning in this study by Max Landsma and colleagues. A follow-up study of the subcortical structures show the effect of progression of Parkinson's on the basal ganglia and the rest of the subcortical nuclei. And then throughout the cerebellum, there's a similar propagating wave of gray matter loss that occurs as the disease progresses. And this is also reflected in DTI, where the microstructural degeneration in this study by Connor Owens-Walton is seen to proceed. In development as well, there are common and distinct patterns that are emerging across disorders. So Sophie Townend aggregated data from their own antisocial behavior working group and also the ADHD, anxiety, and major depression working groups, and found that all of the disorders that are characterized by externalizing symptoms, aggression, impulsivity, hyperactivity, and conduct problems, are associated with surface area deficits, particularly in the frontal cortex. But the more depressive and internalizing symptoms have much more limited effects in the brain. And they published a landmark study comparing all of these signatures, pointing to a common externalizing specific reduction in surface area. This study by Marlene Staginis and Yidian Gao took this further and found profound frontal thinning in youth with conduct disorder across 15 international cohorts. Again, surface area is the greatest brain effect that's been seen in conduct disorder, much more so than cortical thinning. And this led Kuldeep Kumar, Sebastian Jacquemont, and colleagues to note that the earlier the illness has an age of onset, the greater the surface area deficit, whereas in adult onset disorders, there's a more prominent cortical thinning. Now let's look at this disorder by disorder. Haley Wang and Tiffany Ho looked at childhood neglect. They found primarily surface area reductions, but no cortical thickness effects in children who had been physically and emotionally neglected, as assessed by the CTQ. Laura Berner, in a new and remarkable study of people with bulimia nervosa, found primarily surface area reductions, again, in line with its developmental age of onset. As I said, adult onset disorders tend to have greater effect sizes for cortical thinning. So you could say there is a sort of organizing principle here where the early onset disorders, particularly CNVs or neurogenic variants that affect the brain very early, result in surface area deficits, and cortical thinning tends to occur later. A deeper analysis of these brain signatures has been conducted by Micah Hetwer and Sophie Volk, where they identified that the disorder-specific patterns have a shared component across many disorders, a disorder-specific component, and a medication effect, and two main transdiagnostic axes, which they call G1 and G2, capture how the disorders map onto the brain developmental gradients. Now, what does this mean? Now, gradients are a way to organize the cortical signatures in a pattern that proceeds from primary sensory motor cortex into heteromodal association cortex. And here is the group of people that is pioneering this in the ENIGMA gradient working group. They find that because cortical organization follows continuous gradients from primary to transmodal association cortices, there are distinct effects depending on the age of onset of diseases, where the regions of the cortex that are disrupted the most are often maturing around the age of onset of the disorder. And they have been looking at this also in work by Justine Hansen and Bratislav Misic in relation to receptors and connectivity profiles. So let's move on. ENIGMA addiction has been finding primarily frontal cortical thinning in people who abuse alcohol, cocaine, cannabis, opiates, and nicotine. You'll notice this is very similar to deficits that are seen in externalizing disorders. There's been excellent work by Emily Dennis and team harmonizing clinical measures. You'll know that clinical questionnaires often have many items with similar meanings, and they've been finding a deep learning approach using transformers that will help you integrate information across disparate questionnaires. This work by Emma Corley and Dara Cannon uses item response theory to integrate information from disparate questionnaires on symptoms in bipolar disorder and major depression. And this paper accepted today, hot off the press, by Sinead King and Colin McDonald, shows that bipolar patients who take different medications have different brain signatures. They find people taking lithium have hypertrophy, and people taking anti-epileptics and antipsychotics have distinct signatures of brain abnormalities, which has just been accepted today. Now, a similar line of work by Shara Sararans, Leon Schmal, and colleagues has found that serotonin norepinephrine reuptake inhibitors, a commonly prescribed medication in major depression, strongly interact with the age effect on cortical thickness. They're very interested in age by type of medication interactions in terms of brain aging. Let's look at some additional disorder-specific results. This work by Wolfgang Omlor and colleagues found that the variation of the brain structurally is much higher in schizophrenia patients than in matched controls. So they found that by just looking at the variance of the cortical thickness, subcortical volume, and cortical surface area, you are often finding up to 1.5 times higher variance in the patients. Now, this will lead you to think the disease has less of an effect in diseases where the patient variance is higher, which is often typical in progressive conditions such as Parkinson's and schizophrenia. And they also find this for DTI. The schizophrenia working group, led by Jess Turner and Theo Van Erp, have been associating brain signatures with amotivation, aggression, and negative symptoms. Congrats on this new work by Kannan Vijay-Kumar, who's working with Theo Van Erp. You'll see all of the different subdomains of affect have been mapped not just onto the brain, but also onto different receptor distributions. This work by the early onset psychosis group has found similar cortical thinning in early onset psychosis, but greater than adult onset schizophrenia. Here's some of their maps by Stena Nerland, Claudia Barth, and Ingrid Agarch. You'll see that gyrification is a very interesting signature, probably one of the strongest. And there's a great deal of interest from Thomas Matthiesen and others in looking at local gyrification further across psychiatric illnesses. Relatives of people who have a mental illness who are unaffected also have brain signatures that are shared to some extent with the patients. So this work by Marjolein Berendse has found that first-degree relatives of bipolar patients have relatively protected brains. In fact, even better-looking brains than healthy individuals, whereas they found no differences, at least on DTI, between first-degree relatives of schizophrenia patients and matched controls. At the same time, there's also work looking at network-wide thalamocortical features that seem to be altered in unaffected relatives of people with schizophrenia. ENIGMA Anxiety, led by Dan Stein, Nick Groenewold, Nick Ranway, and Janne-Marie Mas-Hogendam, have been studying several disparate anxiety-related conditions that have high comorbidity, such as social anxiety, generalized anxiety, panic disorder, and specific phobia. And this rather remarkable study that was pre-registered by Janne-Marie looked at childhood-inhibited temperament. They, in fact, found that there was not a link between childhood-inhibited temperament and brain structure. This was a pre-registered study. However, a group looking at specific phobia, led by Kevin Hilbert, found that animal phobia and, in fact, blood injection injury phobia was associated with a subtle but consistent effect across the brain. ENIGMA OCD, led by Odile van Heuvel and Dan Stein, have been studying, using a variety of imaging approaches, the prediction of cognitive behavioral therapy outcome, medication effects. They've been looking at the cerebellum, polygenic risk, task-based fMRI, and also a number of future plans are integrating multimodal data in a new R01 project that combines the efforts of another consortium, Global OCD, with ENIGMA OCD to launch larger-scale multimodal analyses. Congrats to Laura van Velsen, who's been studying suicidality. She found that lower fractional anisotropy is associated with a history of suicide attempt over and above the effects of other psychiatric diagnoses. Now, there's great interest in identifying potential markers of suicidality. She also found that recent suicidal ideation was associated with higher axial diffusivity in the cingulum with small but consistent effects. Now, let's turn to neurology. Heath Pardo completed a remarkable study in which he found that the brain age acceleration in epilepsy patients was greater in countries with lower income and greater income inequality. Now, this is a stark reminder that we should look at diseases across different countries. Obviously, if income inequality strongly influences the degree of brain differences that you see, which is the case here in epilepsy, that should also be studied as a potential modifier. ENIGMA Epilepsy is analyzing data from 38 centers, led by Kerry McDonald and Sanjay Sisodia. They have 3 R01s under review, so good luck to them. In Enigma Parkinson's, led by Isbrand van der Werf and his team in Amsterdam, published a remarkable cross-modality study. This was led by Andrew Vo at McGill, mentored by Alan Dagger, in which they found that Parkinson's disease, evaluated using MRI data worldwide, is like a seismic event with epicenters. So like an earthquake, the disease propagates via network connections. And they identified how the disease spread relates to neurotransmitter distributions, cytoarchitecture, and functional networks. And this is a true, what we call Montreal-style paper that integrates a vast host of information. Congrats to them. And also, Zina Shawa has this intriguing and quite influential study revealing 3 subtypes of spatiotemporal atrophy in Parkinson's disease, in addition to the subcortical atrophy subtype that is most well known from the textbooks. They also found a cortical first and a limbic first subtype of atrophy, which is of interest in understanding perhaps differences in treatment response. Ian Harding has been studying in Enigma ataxia the differences in the cerebellum associated with earlier onset, which are usually worse, or longer duration of illness, which are also usually worse. And this new study of disease staging using SARA scores, this is grouping patients by clinical severity, not by atrophy sequence, has identified a staging system for spinocerebellar ataxia type 2, also known as SCAR2, which is very useful for evaluating patients' degree of disease using brain maps as a reference. Enigma chronic pain had an incredible result in recent weeks. So this is led by Sylvie Augustin and Jan Kide with support from Neda Jahanshad at USC. They looked at 36 independent data sets with T1-weighted MRI. And have a look at this. So they do disaggregate pain into nociceptive pain, neuropathic pain, migraine, I mean, many, many types of pain. But when all of the patient's data is aggregated, there's a profound reduction of surface area in people with chronic pain. This is based on over 2,000 people. They find a cortical thinning pattern in a completely novel pattern. This doesn't seem to be identified in any other disorder that we study. And this is cortical thinning associated with chronic pain. And they're beginning to look at the functional consequences using resting state EEG. Enigma Sleep, that's led by Masoud Tomasian, has been having Enigma Sleep webinars. You're all welcome to join. 23 talks are available. Do have a chat with Masoud if you want to listen into those. And they're starting a collaboration with Enigma PD on REM sleep behavior disorder, which is a prodromal condition, Parkinson's disease. There's a monumental study led in Enigma Sleep by Han Wenbi, looking at the effects of insomnia or sleep deprivation on cognition. And anyone with jet lag will know about that. Glymphatic indices after sleep deprivation are of renewed interest in neurology. So the glymphatic system, which is shown here, clears toxins from the brain in addition during sleep, and it is now implicated in Parkinson's disease and a number of neurodegenerative conditions, emphasizing the role of good sleep, which is known to potentiate this effect. Now, there's interest in forming a glymphatic working group. We're thankful to Ho-Sun Kim at USC, who's developed a whole host of tools for quantifying glymphatic function, either using T1-weighted MRI to quantify perivascular spaces, or more in-depth methods such as DTI-ALPS, which look at the integrity of the blood-brain barrier. Let's also look at some new findings. So Perry Pascu, Kevin Black, and colleagues have led the first large-scale study of Tourette syndrome. So following the successful model of Enigma OCD, they followed the same pathway. I think anticipating that Tourette might look a little like OCD. And in fact, the surface area and cortical thinning patterns seem to emphasize the inferior temporal cortex, which is not similar to OCD, whereas there are similarities subcortically. And they found a genetic pleiotropy. It's helpful that Dr. Pascu also leads the PGC working group on Tourette syndrome. They found a genetic pleiotropy between the loci that put people at risk for Tourette syndrome and the major loci driving caudate and putamen volume. So there's a mechanistic explanation for these findings. They also have found profound disturbances in brain asymmetry in children with Tourette syndrome, but not in adults. So that's another interesting new lead. Jan Beutelaar and Kathy Lawrence have been aggregating data from autism spectrum disorder across 27 sites. They've finished a study with structural imaging. They're processing DTI, and they're planning resting state fMRI. And just to get excited about this, they have a number of subtyping, brain gradient studies, even studies of the cerebellum in autism that are leading to promising results. These ones by Gao and Kim, working with Kathy Lawrence at USC, implicate subregions of tracts that are altered in children with autism spectrum disorder. And a related study by Kathy Lawrence implicates gray matter reductions. And given the recent interest in autism, is especially important to find robust biomarkers. ENIGMA 22q, which is a neurogenetic syndrome that puts people at risk for schizophrenia, has been studied by Runa Bowen with Carrie Bearden at UCLA. They found that the deficit pattern in 22q is linked to neurotransmitter receptor density. And this, again, is a cross-modal analysis, which is shedding light, particularly in neurogenetic syndromes that have deletions that affect gene expression. And it's very reasonable to expect a genetic deletion that affects directly gene expression to have cortical anomalies that correspond to that transcriptomic profile. So they've been looking at this study by Jen Forsyth has been looking at normative deviations in 22q and their relationship to other behavioral measures. And they're now beginning a multimodal project. ENIGMA Origins, led by Rebecca Nickmeyer and Alex at Michigan State University, have analyzed infant data from birth to age 6 in 7,800 children. And it's absolutely remarkable. And if you see how many images they're processing, it's no wonder they've published 6 studies this year. They look at the infant brain and have found developmental differences in infancy that are related to maternal obesity and diabetes, antenatal maternal depression, basal cortisol levels. And they've also conducted genome-wide association studies that have found genomic markers linked with resting state functional synchrony in infancy, and even cerebrospinal fluid phenotypes and complement 4 variation, again, all in infants. This study by Fabio Bernardoni, working with Stefan Ehrlich at the University of Dresden, has been looking at anorexia nervosa. Now, anorexia has one of the greatest effects on the brain of any psychiatric condition. The cases tend to have one standard deviation lower morphometric measures than typically developing in individuals. But there is hope. Many of the individuals do recover with weight restoration. Their brain metrics regain normality. So these charts, as you see here with the red individuals being those with anorexia, they're of intense interest in gauging whether a patient undergoing nutritional restoration is improving relative to their prior centiles. They've been doing machine learning and they strongly classify anorexia versus healthy controls, but it's been more difficult, at least for now, to differentiate anorexia subtypes. But they have, in this rather remarkable study, found a link between serotonin serotonin transporter expression and the profile of anomalies seen in anorexia. And I encourage you to check out that if you're interested. They're also beginning in Clara Moreau's lab a study with Fabio Bernardoni of resting state functional MRI in anorexia. Tim Silk, Nanti Vijayakumar, Amanda Boys, and Sylvia Lynn have started, and they're also funded to analyze puberty, endocrine changes, and their relationship to brain development in 35 cohorts. And there are some additional highlights. So let's look at this. So Clyde Franks, Eileen Luders, and Shan Kong have found altered structural brain asymmetry in bipolar disorder, an alteration in lateralization. This follows on work in PNAS showing that the lateralization of the brain in schizophrenia is reduced, which was hypothesized by Tim Crow and Eve Johnston about 50 years ago, and they 50 years later found it was true. ENIGMA Diabetes has emphasized the role of cardiometabolic conditions, systemic conditions, in the worsening of mental health conditions. So Shan Luo, Tamara Hershey, and Eustace Hugh at USC are actively seeking collaborators with information regarding metabolic disorders, and you can see that's going very well. And work by Thomas Hayek and colleagues in Halifax in Nova Scotia at Dalhousie University has led to a series of papers showing that a good part of the anomalies that you see in bipolar disorder and perhaps also in psychosis can be attributed to obesity, metabolic and cardiovascular factors, which leads to a line of work pointing to the value of stabilizing those factors for mental health outcomes. This This new funding to Enigma Stroke, congrats to Sukley Liu for winning a major NIH grant to harmonize stroke rehabilitation data sets. They have now have an Enigma Stroke database. And this has led to very interesting studies by Martin Domen and Martin Lutzer, trying to understand which brain markers predict recovery in younger people with stroke. And they found that in early work, not just the percent of the corticospinal tract that has been infringed on by a stroke, which is a very common measure of the severity of stroke, but also the FA in different parts of the brain, ipsilesional parts of the brain, is helpful in predicting recovery. And then taking this even further, Gilson Park and Ho Sung Kim have a paper in Press Atlantic Digital Health where they look at local brain age. So local brain age is how much older does your brain appear to be than your chronological age. And in different parts of the brain that are ipsilateral to the stroke, they found that this was extremely useful, in fact, much better than standard metrics in predicting motor recovery in stroke, which this is a very practical system to give some indices of prognosis in people who are recovering from stroke. They have a whole host of very interesting secondary analyses looking at corticospinal tract lesion load, sex differences, post-stroke depression, and a number of tools which are being brought to bear on their database. Emily Dennis, Alex Olson, and the moderate to severe traumatic brain injury working group have been looking at both acute and chronic effects in groups of people with TBI and in individuals with TBI, showing that normative modeling, again, has value. Many of these neurological conditions, brain trauma and stroke, often have good recovery, and they're using the imaging at baseline to predict what that recovery should look like. And there's been renewed interest in projects in the intimate partner violence subgroup of the TBI working group, and also new work by Jacqueline Caucase and Hector Arciniega on sports-related TBI, primarily concussions and sports-related injuries. Lauren Salmonen, Megan Herting, J.C. Chen, and Teresa Monreal have been geocoding brain data. So they, if you ask them, will index your brain scans and bring in a whole host of pollution data, access to healthcare data, other environmental measures, for example, income inequality. And if you didn't look at these, you'd never know they were affecting the illnesses that you're studying. So they'll help you map those. And then finally, this is the very last update. Sampras Ganasan from Melbourne University and Tony King at Ohio State published an incredible review. I encourage you to read this on all the brain imaging studies of meditation. They're a very, as you would imagine, chill group, very enjoyable to work with this group. And many of them do meditate themselves and are practitioners of it. And they'll tell you about how meditation has been effective in at least a subset of patients with major depression, PTSD, and other types of trauma. There have been a number of educational initiatives. So last 2 minutes, we'll talk about some of these. There's an effort led by Rian Gi, Shalala Haas, supported by Sophia Frangu, called the Translational Neuroimaging Educational Program. This is online. You can learn about all sorts of brain imaging techniques, show your students them. It's a very interesting set of methodological talks, including one by Riyang in Chinese and in English, which is quite remarkable. Emily Dennis has developed something that is quite extraordinary. So Enigma U is a free platform in which students, now nearly 2,000 students from 98 countries, are able to learn about the brain, neuroscience, psychiatry, careers in science, particularly in under-resourced parts of the world where people just simply wouldn't have access to any type of neuroscientific training. They've moved this to Google Classroom, which is free. There's no enrollment cap. If you'd like to contribute lectures, and many of you have, do have a chat with Emily. They're extremely interested in these lectures. They're very, very popular. They're only about 15 minutes long. And many of you, I mean, we have this incredible landscape of expertise here. There are now 176 lectures, over 57 hours of training materials in Enigma U. This very practical course, 2-day conference on intimate partner violence, was attended by 750 people. So Carrie Esopenko and her colleagues have organized this every year, and you will be able to see last year's recordings on YouTube and this year's fairly soon on clinical and research questions in intimate partner violence. We hope to see you in New York in April, or even better, in Bordeaux in France in June. So you have 11 hours to submit your abstract to the Human Brain Mapping meeting and come and see us there. We'll probably have more than a social event at those. We'll probably have a full-day workshop in New York and maybe a full-day workshop for those of you attending the OHBM, partly because some of these newer methods, the resting state functional MRI, the tractometry that we talked about a little bit earlier, and all of the normative modeling methods, I mean, they're yielding such extraordinary findings that I think a lot of us would like to learn from each other about what the most promising results have been. And then also a big thank you to Nadia Ginelia and Connor Owens-Walton, who've really been exceptionally supportive to ENIGMA OCD and ENIGMA Parkinson's disease, respectively. They're now moving on to new ventures in their own careers. So a big thank you to Nadia and Connor. And then last of all, if you're not getting any emails from us, do tell Sophia. All of you should be on the ENIGMA listserv. There's also some job opportunities that you can take a look at. But if you're not getting emails, tell Sophia. And just last, a word of thanks. So huge thank you to our USC team. Specific thanks to Sophia Thermopoulos, who's our air traffic controller and helped create this presentation today. In fact, many of you contributed remarkable studies to it. Thank you all for making ENIGMA what it is. Thank you for your leadership. Thank you for your determination in the face of a difficult funding environment. And again, thank you for those of you who at the beginning paid tribute to Professor Dan Stein and his remarkable impact on global neuroscience, and we will miss him. So thank you again. This was a great scientific year. It's even better now we've finished this, so we don't have to worry about it. And I think the instruction is to turn on our cameras for a group photo. So let me see if I can get that right. I think it's wonderful to see everybody. And in fact, what a remarkable meeting. This is so nice to see everybody. So I don't know if anyone has final remarks. I doubt that we ever are present in these numbers. I mean, we wish everyone a restful holiday. For those who've had a difficult year, let's hope for a better one next year. And I think we can look back on some of the major scientific achievements and look forward to a very, very exciting future.
Thanks, Paul, for being such a great leader, bringing all these people together. It's really, really great. Thank you.
Thank you, Adil. And wish you all a wonderful holiday and hopefully a better year next year, an even better year, or at least a good year next year.
Thank you, Paul.
Thank you, Paul.
All right, cheerio for now.
Thank you so much, Paul. Thank you, everyone.
Bye.
Thank you, everyone.
Bye-bye.
Wonderful.
Thank you.
Bye bye. Nice holidays.
Happy holidays.